Okay, hello, my name is Atena, and my project is on investigating the relationship between the skin microbiome and acne vulgaris. I'm pretty sure that you or someone you might know suffers from acne, and I have dealt with acne since middle school, which is why I wanted to do this project. So acne is one of the most common skin conditions around the world, affecting more than 85% of adolescents worldwide. And it is an inflammatory skin condition caused by blocked phytosebaceous units, which is also known as the hair follicles. And this is often followed by tender red bumps. Although acne does not have an adverse impact on a person's life, nor does it impact the overall health of a person, it can have an adverse impact on the individual's mental health, and it can be compared to depression. Despite its prevalence and its impact on a human being, we still do not know the exact causes of acne. Fortunately for us, recent studies have discovered that the skin microbiome plays a substantial role in the development of acne. And one of the bacteria that they identified was Cutibacterium acnes. And a lot of the treatments today relating to acne actually works to eliminate Cutibacterium acnes. So what is a skin microbiome? So the skin microbiome is all of the microorganisms that live on our skin, including the bacteria, fungi, and viruses, and it can be split into two different types, the resident and the transient. So the resident skin microbiome refers to all of the more commensal bacteria that permanently lives on our skin and helps to protect it, while the transient microbiome is affected by the external environment and is more temporary. So knowing that the skin microbiome may have a relationship with acne, I came up with two research questions. So first was, are there differences in the phyla, gender, and the species between the skin microbiome of acne-affected and healthy individuals? And two, what is the prevalence of Cutibacterium acnes among acne-affected and healthy individuals? So through my project, I wanted to find and I wanted to have a better understanding of the skin microbiome in relationship to acne, so that in the future we can come up with more personalized skincare products and therapies for patients, because not all remedies at the moment work for everyone. In order to investigate this, I used a 5-step procedure in which I recruited, sampled the skin, extracted DNA, and conducted PCR, sequenced the DNA, and analyzed my data. So the first step was recruitment, where I recruited 2 acne-affected and 2 healthy individuals based on the global acne severity scale. And so I made sure that the normal individuals, the healthy individuals, did not have any sign of acne vulgaris and were in the grade clear, whereas the 2 acne-affected individuals were in the moderate area, where there were few non-inflammatory lesions and maybe one cystic acne lesion. So in order to collect the skin samples, I decided to use a skin swabbing method because it is the most non-invasive method when compared to methods such as skin scraping. And I wanted to make sure that my methods were optimized. And so I compared 2 different types of swabs. So the first one was swab moistened with saline solution, and the other type was an e-swab. And when comparing these two, I found that the e-swab collected more DNA, and I decided to go with the e-swab. So the morning of the skin sampling, I made sure that the participants did not wash their face, so that I can preserve the most natural skin microbiome as possible. And these participants were swabbed using the e-swab. And once we got that sample, I extracted the DNA using a pyrosoil kit. And the reason why I decided to use a pyrosoil kit was because the pyrosoil can extract even the smallest amount of DNA, and it was also used in previous research papers that dealt with the skin microbiome. So after we collected the skin samples, I amplified the 16S rRNA gene using the primers 27 forward and the 1492 reverse. And the reason why I decided to look at the 16S rRNA gene is because this is a highly conserved region in bacteria, and we can use this to identify the different bacteria species in the samples. After the amplification, I cleaned up the DNA that we had using Ampere beads and barcoded each sample for identification when we sequence. Finally, these samples were loaded onto Fongle and sequenced using the Oxford Nanopore MinION, and these sequences were blasted on the Epitome from Nanopore program. Once we got the data, I decided to look at it on 3 different levels to get a different snapshot of the data. And so I first looked at it on the phylum, then the genus, and then finally the species. So if we take humans as an example, the phylum would be Chordata, the genus would be Homo, and the species would be Homo sapiens. So as we can see on the stacked bar graph, this is a data on the phylum level, and we can see that there was no great difference between the skin microbiome of acne in healthy individuals. And this was a result that we expected because we only looked at 4 categories within the phylum level, and it is not very specific, so we did expect no great difference. So for each level, the statistical analysis that I decided to use was cluster analysis and the principal component analysis. And the data that we can see here, or the analysis that we can see here, is the cluster analysis. And what the cluster analysis does is it groups different items based on how similar those items are. So the closer they are together, the more similar. So on the phylum level, we can see that normal 2 had the greatest difference from all the other samples in the cluster analysis, and it is highlighted in yellow. On the principal component analysis, we can see that the acne 2 had the greatest difference. And what the PCA, or the principal component analysis, does, it shows why something has such great variance. So we can see here that acne 2 had the greatest difference because it diverges the most from all the other samples. And one thing to note is that the cluster analysis and the principal component analysis are quite inconsistent at the phylum level, which is why I decided to also look at the genus level. So on the genus level, you can see straight from the stacked bar graph that acne 2 seems to be an outlier, and we do see a lot more, we do see that the data does vary a lot more on the genus level. And we can see that acne 2 seems to be an outlier because of the lactobacillus, which is in green. As expected, the cluster analysis also showed that acne 2 had the greatest difference. And likewise, in the principal component analysis, acne 2 had the greatest difference due to the lactobacillus. And so on the genus level, the cluster analysis and the principal component analysis actually were consistent. Because we did notice that lactobacillus isn't really a bacteria that is related to acne, and it had quite an impact on the genus level, I also decided to look at the species level. And when I looked at the species level, I only selected for bacteria that was already known to have relationship with acne. And so these are definitely not all the species that were found, but it's the bacteria that is heavily related to acne. And so on the species level, we can see that all samples contain Cutibacterium acnes. And as I've mentioned before, it was previously known that Cutibacterium acnes was the cause of acne, but we can see here that all the normal and the acne samples had Cutibacterium acnes. So one thing that we found that was really interesting was our results in our principal component analysis on the species level, and here we can see a bit of a pattern. So here we can see that acne 1 and acne 2 have the greatest variation. So we can see that acne 1 and acne 2 differentiate from where the normal samples are. So here we can see that there is a great difference between acne 1 and acne and the normal samples. And one thing to note is that acne 1 is pulled by the bacteria called Staphylococcus, while acne 2 is pulled by the bacteria Cutibacterium acnes. And from previous research, Staphylococcus and Cutibacterium does have a role in acne formation. So from my research, I was able to find that there's no great difference between the skin microbiome of acne-affected and healthy individuals until the species level. So excluding acne 2, the skin microbiome composition was quite similar between acne and healthy individuals on the phylum and genus levels. However, starting from the species level, we see that acne 1 and acne 2 can be differentiated from normal because of the presence of either Staphylococcus or Cutibacterium. And when we go back to our genus level data, we saw that there was a dominance of lactobacillus in acne 2. And I was wondering why this may have what happened, because lactobacillus is not present on the skin at such high levels. And I did find that the participant that we got acne 2 from actually did use a probiotic product on the skin prior to sampling. And this makes sense because lactobacillus is a probiotic bacteria genus. So overall, my research may suggest that acne is not caused by an overabundance of a single bacteria. As we saw, all samples contained Cutibacterium acnes, and there was no quantitative difference between the C. acnes proportions. And therefore, treatments that aim to eliminate one type of bacteria may not be as effective. So through my research, one conclusion that I can make is that the presence of Cutibacterium acnes is not the only cause of acne, and future treatments should look at treating the entire skin microbiome as a whole, not only working to eliminate Cutibacterium acnes. Of course, my research did have its own limitations. The first thing was that the sample size was quite small. We only had 2 acne-affected patients and 2 healthy individuals. And number 2, the research was conducted in the same laboratory as other researchers studying oral and vaginal bacterial microbiomes, and we did see some contamination in the data. And number 3, we were unable to keep the skincare products or the external environment constant for all the participants. And I would like to conduct further research from here and look at the different types of phylotypes and dive deeper than the species level, and also conduct the same research in a different region because the skin microbiome is known to be heavily affected by the external environment. Some advice to future students who want to take honors, independent research in science is to not be discouraged by no results. I did have times when I did all this work and no results came up, but sometimes having no results is a result itself. And in times of failure, do not give up. I definitely felt this when I was optimizing my methods, because I spent quite a lot of time optimizing my methods because I wanted to make sure that my methods were the best it could be. And I would advise only to research what you're truly interested in, no matter what anyone says, because you do have to stick with it for a full year. And what I learned through this experience was that was how complex studying the microbiome was. I already knew that it was quite complex, but I didn't know how much work was put into it until I experienced it myself. So I would like to thank all of the people who have helped make this project possible. This research would not have been possible without Mr. Hill, Mrs. Kobayashi, and Dr. Chan, who have helped me along the way, and all the volunteers who have participated. Thank you so much.